Good. Hi, everyone. Thank you for attending this workshop. You are in the workshop for building a general inference engine for molecular structures and ensembles. I'm one of the organizers. I'm Doris. I'm a postdoc at SLAC working on machine learning method development for crystallography. And the two other organizers for this workshop are Minhuan and Aldama. You guys want to briefly introduce yourself?
Yeah, I'm currently a postdoctoral at Flatiron Institute working on the inference problem and also like all the Bayesian inference techniques.
Hi, I'm Luis. I'm a grad student at Harvard University, and I work on machine learning methods for crystallographic data analysis.
This workshop is really a continued series from last year's workshop initiated by Kevin Dalton and Minhuan. And we receive a lot of support from Frederick Poitevin, who is leader of the machine learning and computer vision group here at SLAC. We're doing this workshop because we're in a very exciting time of structural biology. So after decades of attempting at this protein folding problem, we have some pretty amazing success from machine learning method, especially AlphaFold that really made its name even in the general public. And right now we're on AlphaFold 3 that not only does just protein structure prediction, but can also do prediction on complexes, protein-protein complexes, as well as nucleic acids, also like binding to small molecules. It's a pretty generic prediction on biologically relevant molecules. And we're obviously not done here. This amazing success from machine learning has inspired us to look at beyond just a kind of a single structure prediction. And that's illustrated by some of the like even newer methods that come out since then. For example, Bolt 1 and 2 try to incorporate some of the conditioning strategies to incorporate like additional information, as well as molecular dynamics information to specifically improve the binding affinity prediction between protein and small molecules. And BioEmu tried to kind of elevate this notion of dynamics even further, where the objective is no longer to like we predict one single structure, but we're actually trying to learn kind of an equilibrium distribution, a thermodynamic distribution of structures that can best explain the experimental observable on a kind of macroscopic level. And meanwhile, on the experimental technique side, we also have a lot of developments going on that includes both kind of the throughput, we have more data coming in, but also on kind of the quality of data that we're getting. So I'm going to talk on the crystallography side because that's what I work on and here at SLAC. So for example, here at LCLS, we're upgrading to HE, which means that we're getting X-ray beam that are not only like brighter, but also deliver on a much higher repetition rates. And that would enable us to look at time-resolved structures to probe the dynamics, especially like non-equilibrium dynamics under various kind of perturbation, whether it's like small molecule binding or temperature or other kinds of perturbation. And other than this kind of like serial crystallography, well, there are also other very exciting developments going on just on kind of the data sets front. For example, Diamond Light Source just got this kind of 8 million investment to do this open bind, which is essentially a data set specifically for AI ML development on small molecule drug discovery. So that's curating this kind of fragment screening data sets between like protein and small molecule binding. In addition to X-ray crystallography, cryo-EM is another like very exciting field. As you can see from this figure, where like over the years we have kind of increasing adoption of this technique as well as method development that kind of further improve this technique. So you can see that we have like increasing number of density maps deposited using cryo-EM, as well as kind of pushing towards higher and higher resolution. And at this point, it's not that uncommon to have near atomic resolution from cryo-EM data. And we can even start to model like ligands into these cryo-EM maps. And obviously there are like other techniques, like for example, NMR, solution scattering, and single molecule scattering that are well positioned, perhaps better positioned to capture this kind of dynamics and like ensemble information. That I personally don't have a lot of background in, but hopefully we can hear from some of the speakers today. on some of the like exciting progress in these fields. But the point here really is that we're having this consensus on this paradigm shift that will happen from just standalone average structure prediction to incorporating dynamics, at this point probably equilibrium dynamics, and ensemble information. And we see that happening both on the experimental technique side as well as on the computational modeling, especially machine learning efforts. And this means that we're trying to, like, as I said, like move away from this modeling single average structure, but we're trying to like infer perhaps a structure that's more relevant to your experimental observable, and maybe infer maybe a collection of structures, a distribution of structures that are relevant to your experimental techniques. And Minhuan will take over to talk about what specifically this inference problem is, mathematically how that maps to the specific details of each experimental modality, what are some of the shared challenges that, that we hope to address in this workshop.
Yeah, thanks, Paige. So basically, like, even though today our audience and speakers are coming from different experimental modalities, we're still trying to put the whole like inference problem into a shared framework, so we can think about like what is the problems and challenges we share as a common community, and then we can work on in the further effort as a community together. So basically, like if we try to like write down this like very simple like Bayesian equation to write down like what our target is, basically the posterior distribution, whether we're trying to doing the sampling for it or doing the point-wise estimation for it. It's basically like we have some experimental observables coming from different experimental modality, and we're trying to sample like what is the biomolecular states we're interested in given that observables we have, right? And we try to dive into specific terms. First, the likelihood term, especially for the biomolecular experiments, like the likelihood term can sometimes be expensive or intractable, right? Because like there are so many different contributions, which can be like hard to physically modeling in your formation of your signal. For example, like like in crystallography, we have the bulk size contribution, which also contribute a lot to your resulting signals, but it's hard to have a very accurate modeling of that so far. And also like specifically like for the experimental modality, well, like the signal is sparse and the noise comes out very strongly, like the single molecular scattering. That's definitely what tells us. It's kind of important to explicitly account for the different noise sources, and how you can model that with a very accurate physical equations can be actually important. And more importantly, like in the car-EM sense, sometimes you have to take into account these like nuisance parameters, which actually not super important to the biomolecular states you're interested in, but like they're important for the likelihood calculation, like the pose, like the CTF parameters. Imagine like you have 100K single particle images in car-EM. You have to infer these nuisance parameters for every single one of them, and imagine how expensive that is. And all this expensive and intractability of likelihood can point us to different techniques about how you can solve that. And more importantly, we have this differentiability requirement for the calculation, because in the following, if you want to like connect this with like the machine learning based approach, you will need some differentiability or gradient assistance in your sampling. And we also have the priors, which I think is actually the most interesting part of the biomolecular sampling problem, because we have most of the biomolecules actually super constrained. They only live in a very specific subset of the high dimensional space. They're constrained by the energy, right? And the good news, we already have a good history of development in the prior side. We have the many like physical priors coming from the MD side. We can also use very general like arguments like the smoothness priors used by relying people. And we also have like these existing samples coming from anywhere, like we can use as a bias samples to initialize our sampling. And now more excitingly is like we have the many like pre-trained models like AlphaFold2, like all the diffusion models in proteins we can use as pre-trained foundation models to help us samples, right? It's like on one hand, it's like actually like gives us a good starting point for the sampling problem. On the other hand, it's also like add an other layer of the complexity in the sampling pipelines. So we'll hear about like some efforts from our researchers today. And more importantly, it's more like a scientific problem. It's like even you have the very accurate point-wise estimation of your px and your likelihood py given x, it's still pretty hard to sample from this px given y posterior, because it's lived in a very high dimensional and multimodal distribution. And how to sample from that using an unnormalized pointwise estimation is highly untrivial. Imagine like you're trying to sampling from something like this. It's super multimodal, it's also super rugged. And it's even worse than what we demonstrated here, because like the valley part between the peaks in the real posterior can actually be very rugged. So your signal, your gradient signal can actually be totally lost during that transition states. So the good thing is that we have like many existing like inference machinery from the statistical part. Like we have the MCMC. We also have like people working on the generative modeling part to solve these problems. And we have the new like paradigm, like the diffusion model guidance. And there's also a new like whole new field in statistics called neurosampler, which we try to use all the neural network-based method to help us sample from our normalized energy function. And then we'll try to help out all the exciting development today. And this is our schedule today. We have speakers coming from very diverse experimental modalities, all working on these inference problems. We have Stefan working on the single molecular X-ray scattering. We have Sujong working on these like protein stability data sets, also working on the bioemu project. We have Mark working on the cryo-EM, and also Chen Chen also working on the cryo-EM. We have Mikhail working on the solution scattering, and Stephanie and Asanka is working on the general ensemble sampling problems and also the crystallography data sets specifically. Okay, I will leave the talk to our first speaker, Stefan.
Yeah, thank you very much.
Yeah, just very briefly, we will have a coffee break and workshop photo, so please take the photo with us. And then for the speakers, we have about 25 minutes for the talk, and then we'll open it up for 5 minutes of questions. I'll be sitting right there, so I'll probably just give you like a hand sign that you have 5 minutes left. And then maybe Minhuan can just, but maybe you can message any Zoom speakers to let them know that they have 5 minutes left.